Okay, we'll wait for 2 minutes more for joining of the students. Then we'll continue the class. Is it visible to everyone? Screen visible होଉଛି ସମସ୍ତଙ୍କୁ? Okay. So today we will discuss about the redox reactions and electron transfer reactions occurs particularly in by which mechanism the electron transfer or redox reactions occurs in transition metal complexes, particularly transition metal complexes. Previously, you studied about the substitution reaction in octahedral complex and the square planar complexes. But when a substitution reaction occurs, you can check there will be no change in oxidation state of the initial metal. Suppose you are taking platinum Cl4 complex and in the final product you are getting platinum 2Cl and 2 bromine, but the platinum's oxidation state it remains consistent throughout the reaction. Particularly in the substitution reaction, there will be just bond making and bond breaking occurs. There will be no change in oxidation state of the metal. But here in electron transfer reaction, in some cases there will be no bond making, bond formation occurs. Just simply the electron transfer occurs from one part of the complex to another part. So when I'm saying electron transfer occurs, definitely from one complex electron will flow to another complex, and the complex which loses the electron, I can say this is called as a reducing agent. This is called as a reducing agent, and the complex which gains electron, I can say this will be the oxidizing agent. Or simply you can read out here the complex which loses electron. Here the oxidation state of metal increases and it is said to be oxidized. Or I can say this will be a reducing agent, or I can say this will be a reductant. And on the other hand, the complex which accepts the electron or which gains electron, the oxidation state of the metal will decreases and it is said to be reduced, and the complex is called oxidizing agent, or I can say this will be oxidant. So particularly the electron will flow from lower oxidation site to the higher oxidation site. Here in the initial complex, you can check the star mark. The complex is having the star mark. Here, the oxidation state atom is plus 2, and here the oxidation state of Fe is plus 3. So when an electron will transfer from this complex to this complex, the product will be just like that, FeCN6 3 minus. That means here the oxidation state of Fe is plus 3, and here the oxidation state of Fe is plus 2. Okay, that means electron will transfer from this one to this one. And the electron flow always occurs from the reductant side to oxidant side, or I can say from the reducing agent side to oxidizing agent. So here the electron transfer process, it can occur by two mechanisms. One is called outer sphere mechanism, another is called inner sphere mechanism. So first we will discuss about the outer sphere mechanism. So in outer sphere mechanism, for transfer of electron, the oxidant and reductant need to come closer to each other. That means the two coordination spheres, I can say one is reductant, another is oxidant. It must be come close to each other, or it must be in a condition just like intact with each other. Or they can, I can say the two coordinating spheres are just like joined with each other. That means the difference in between the two coordination sphere, it will be very very less, or the bond difference, or I can say the space difference in between the two coordination sphere, it must be very small, or I can say the two coordination spheres must be intact with each other. So when the two coordination spheres are occupy a nearer position, then the transfer of electron takes place from reductant to oxidant. Then how this electron transfer occurs, we can check step by step. So in the first step, the oxidant and reductant, it needs to come closer and form a precursor complex. So in the two coordination sphere, OX means oxidant complex, RED means reductant complex. So when the two complex come close to each other, they will form a just like a precursor complex like that. And in the second step, there is activation of the precursor complex, which includes deorganization of the solvent molecules. That means he is saying that before the oxidant and reductant need to come close to each other, oxidant will be surrounded by some solvent molecules and the reductant will be surrounded by some solvent molecules. And there will also a interaction between the oxidant and reductant, some electrostatic force of interaction. So if the oxidant need to come closer to reductant, then it must be overcome the solvent interaction around them. So when the oxidant and reductant need to come close to each other, that means it needs a activation energy to make a intact position with each other. So when the two oxidant and reductant need to come close each other in this position or in this time, there will be some reorganization of solvent molecule occurs. When the reorganization occurs, there will be an effect on the ML bond length. The ML bond length will affects when the electron transfer occurs, particularly when the electron transfer occurs. So in the second step, reorganization of solvent molecule occurs along with the oxidant reductant need to come closer to each other, and there will be transfer of electron from reductant complex to oxidant complex. The reductant complex side to oxidant complex side to electron flow karibo. There will be a flow from this side to this side. So after loss of electron, the reductant complex, it will get a positive charge, and the oxidant complex, it will get a negative charge. And in the final step, the ion pair is dissociated. These two ion pairs, it will dissociate to form OX minus and reductant plus. That means the electron transfer will occur from this side to this side, from reductant side to oxidant side, and there will be a decrease in, there will be increase in metal oxidation state of this complex occurs, and there will be a decrease in metal oxidation state of this complex occurs. That means I can say this will be a positive complex and this will be a negative complex. It 4 minus thila it कौन होला 3 minus that ताମାନେ एटी कौन होला positive ହେଲା एटी 3 minus thila एटी 4 minus ହେଲା ताମାନେ एटी कौन होला ओटा negative addition होला. So three steps. First step is called precursor complex step. Second step is called reorganization of solvent molecule, and here that electron transfer occurs, and there will be change in ML bond length. So why this change occurs particularly? So when there will be change in oxidation state, definitely there will be change in bond length occurs. Can I say Fe2 plus and one chlorine bond length, it will be similar to Fe3 plus chlorine bond length? So we cannot say like that. When the oxidation state or positive charge density on the metal will be more, it can closely attract the other ligand towards itself. When the oxidation state decreases, definitely the ligands it will attract in a less amount comparing to the higher oxidation state. So in your study book also, these types of things are mentioned. Here we can say outer sphere electron transfer reaction R is the reductant, O is the oxidant. They will first need to come each other, need to they need to first come nearer to each other, then they will form a precursor complex. Then this precursor complex it will pass through a transition path to form a successor complex, just like that. R plus O minus, that means in this step already the electron transfer occurs. And the next step there will be breaking of R plus and O minus. And these two are the products. Okay. So in outer sphere mechanism, there is a direct electron transfer occurs from the reductant side to oxidant side. Okay, keeping the coordination sphere intact. So in outer sphere mechanism, there will be no bond making occurs, there will be no bond breaking occurs. Just simply the two coordination spheres come close to each other, and from reductant side to oxidant side, there will be transfer of electrons. And briefly, these things are mentioned. In first step, what occurs, there will be oxidant reductant precursor complex formation, and there will be ion pair formation, and in the last step, there will be dissociation occurs. So in your unit 6 material of MCH-16, you can get all these things. And this is the coordinate diagram or energetical diagram of this particular reaction processes. You can see here, this one will be the reactants energy. It will pass through a transition state to form a precursor complex. Again, precursor complex, it will pass through a transition state to form a successor complex. Then again, successor complex means the ion pair complex. It will pass through a transition state to form the product. So this is typically the reaction coordinated diagram of the outer sphere mechanism. Yes or no? Then we will discuss one by one its examples. So first thing, what are the characteristics or what are the silent features of the outer sphere mechanism? So in first point, it is mentioned that both the reactants should be kinetically inert. If the two reacting complex, one may be reductant, another may be oxidant, it should be kinetically inert. However, if one complex is labile, the other inert complex it not possesses a donor atom, which can be used to form bridge with the labile complex. The second point is not necessary here. It is describing about the inner sphere mechanism. That means the first point is important. Is saying that both the reactants should be kinetically inert. Then the electron transfer occurs. If one complex will be labile and the other will be inert, तो से कुछ गोटे labile होगा गोटे inert complex होगा, and जब गोटे inert complex रो donor atom थिबो, then it can act as a bridging ligand. When a donor atom act as a bridging ligand, then the mechanism doesn't follow outer sphere mechanism. It will be inner sphere mechanism. So after 1 hour, we'll discuss about the inner sphere mechanism. The first thing, when the two reactant complex or the oxidant and the reactant, it will be kinetically inert, then definitely there will be easy flow of electron transfer from one side to other side occurs. So in first complex, in example, he is taking one FeCN63 plus complex, he is taking another chromium water 6 complex, or reduction of FeCN6 by chromium water 6. Okay, he's saying that if both the oxidant and the reductant are labile, there is a possibility of electron transfer from pi star to pi star of oxidant, then the reaction proceeds through outer sphere mechanism. That means he is saying that in this particular complex, these two are the labile complexes. You can check the two are the labile complexes. And what will be the electronic configuration of these two complexes? Suppose I am saying Fe water 6 2 plus. That means what will be the d electronic configuration of Fe here? 2 plus means d6. So d6 means its electronic configuration will be d2g4 and eg6. d2g4 eg2. Here the electronic arrangement will be d2g4 Fe3 plus. So Fe3 plus means d electronic configuration will be d5. If it will be d5, then the structure will be d2g3 eg2. d2g3 eg2. It examples ಬರೋಣ. Just wait for 1 minute. Here you can see Fe water 2 plus, here Fe water plus 3. These both two are the labile complex. Both two are the labile complexes. Here Fe plus 3, T2G3 Eg2. When there will be Fe plus 2, the d electronic configuration will be just like a d5, d6, Fe plus 2. And the electronic configuration will be T2G4 Eg2. That means it's saying that the postulate is predicted that the electron transfer occur from one complex to another. Then tell me, electron transfer will occur from this complex to this one or from this one to this one. So this slow oxidation state, so the electron will transfer from this side to this side. So particularly, the electron will transfer from the T2G4 to T2G3. So when I am mentioning T2G, so T2G means we can other sense I can say this is a pi star orbital configuration. This is a pi star orbital configuration. When I am saying Eg, that means we can represent just like it is a sigma star. So here in electron transfer, pi star doesn't mean antimony orbital. Pi star means it possesses pi orbital symmetry, which is similar to T2G orbital symmetry. That means T2G orbital can make an interaction to form a pi bond, and particularly the Eg orbitals, they are forming the sigma bond. Okay, so in other sense, I can say in place of T2G, I can say pi star. That means here electron will transform from the T2G orbital to here T2G3. That means electron will transform from this pi star orbital to this pi star orbital. So here the transition, I can say pi star transition. That means from T2G to this T2G. Then it will form these two complexes. Okay, you can check the structure again. Here is also labile complex. Here is Also a labile complex. Here is a high spin. This is also high spin Fe2 plus. So water is not a comparatively stronger. It is a moderate type of ligand or sometimes weak ligand also. So the two complexes will be low spin complex, a high spin complex. Okay, that means electron will transform from pi star reductant to pi star oxidant. Okay, that means here outer sphere mechanism, the electron can transfer in between two inert complexes also. It can transfer in between two labile complex also. It can also transfer from one inert to another labile. But when one complex will be labile, another will be inert, you need to check whether the inert one will contain a donor atom or not. If the inert one will contain a donor atom, then it cannot follow outer sphere mechanism, it can follow inner sphere mechanism. In second part or in second example, we'll discuss about when both the reactants are inert with respect to ligand exchange. So one of the reactant will be FeCN6 4 minus, another ligand will be FeCN6 3 minus. Okay, so when the two complexes or two reacting complexes are inert, then the two metal complex will make a close intact with each other and through tunneling. Tunneling means same procedure. They will make a precursor complex and they will make a, just like a tunnel in between them, and the electron will transfer from one coordination sphere to another coordination sphere. Okay, so when the two complexes will be inert, definitely here the electron transfer process it will be little bit faster. Okay, so in outer sphere mechanism, the postulates predicted that the electron transfer always from T2G orbital to another T2G orbital or electron transfer from pi star orbital of reductant to oxidant is faster. It is a faster process. I mean, electron transfer in which types of electronic transfer is feasible? Pi star to pi star electron transfer or T2G to T2G. Because pi orbitals, these are not shielded from the ligands. Pi electron density, it is not shielded by the ligands. But when I am saying sigma orbital or sigma electron density, it is shielded by the ligands. So the sigma sigma transfer, electronic transfer, it is not a faster electronic transfer, but the pi star electronic transfer or the electron transfer from T2G orbital to another T2G orbital, it is a faster electronic process. You can read out here. These are the theories. And with the electron transfer process, if in complexes pi acceptor ligands are present, then the electron transfer process will be faster. Here we can see in cyanide, this is a pi acceptor ligand, this is a pi acceptor ligand, but water is not a pi acceptor, this is a pi donor. So when a pi acceptor ligand is present, it can easily make condition by which electron can transfer to each other. That means pi acceptor ligand means it can easily abstract the electron from metal also. Yes or no? So just like a withdrawing tendency. So when a biaxial ligand is present, it can easily abstract or withdraw the electron density from one complex to another. So you need to remember that in presence of pi acceptor ligand, the electronic transfer process is faster. So comparing to this complex, in this complex, the electron transfer process will be faster because here pi acceptor ligand is present and along with these two complexes are inert. So you can check another example also. So when the electronic transfer occurs by using the same metal complex, here is also manganese present and here is also manganese present. Then these types of reactions are called self-exchange reactions. When the similar types of metal complexes are present, we can say this is a self-exchange reaction. When two different types of metal complexes present, you can check here. Here is iron atom present, here is molybdenum atom is present. Then these types of reactions are called cross reactions. This is called cross one. Okay. So the previous one in point 2, FeCN4, FeCN6, 3, we'll discuss here. how the electron transfer process occurs. So when there will be presence of a strong ligand cyanide, then definitely the complex will be a low spin complex, and it is also a low spin complex. So here the electronic configuration will be T2G6 Eg6. That means in iron, here in iron, the oxidation state is +2. So when oxidation state of iron is +2, in d electronic configuration, 6 electrons are present. And this 6 electron, it will occupy T2G6 position. And this will be Eg0. Here you can check, here Fe plus 3, that means the 5 electrons, it will occupy the d electronic configuration. And this 5 electrons, it will occupy the T2G position. So here the electron transfer occurs from this T2G6 to T2G5. You can also check before electronic transfer. Here the oxidation state is +2. That's why Fe2 plus cyanide bond length is 195 picometer. Here Fe3 plus cyanide bond length is 192 picometer. So in which case the bond length is shorter when the oxidation state will be more. So both the complexes are inert. When both the complexes are inert, the electron transfer process, it will be faster. Additionally, here the pi acceptor ligands are present. Yes, Anu. So the electron transfer occurs from T2G of FeCN6 4 minus. That means from here, from Fe2 side to Fe3 plus side. Electron transfer occurs from T2G of FeCN6 4 minus to T2G of FeCN6 3 minus. That means electron transfer occurs from T2G6 to T2G5. Okay, you can see here pi acceptor ligand is facilitates the electron tunneling process. These things are mentioned here. You can take two high spin complex. In first example, when I am saying T2G Fe water 63 plus, Fe water 63 plus, these two are the high spin complex. Two moderate ligands are present, water water. That's why this will be high spin, and this will be high spin, and the two complexes are labile. You can check initially the bond length is 78 picometer in case of Fe3 plus, and in case, sorry, this one will be Fe3 plus, and here this one will be Fe2 plus. There will be a printing mistake, and here the bond length is 92 picometer. But what is the difference in between the two complexes? In the previous complex, the bond length difference is very less. One is 195 picometer, another is 192 picometer. But here the bond length difference is nearly about 14 picometer. So when the bond length difference will be larger, that means these two complexes, the different Fe3 plus ions orbitals energy and Fe2 plus ions orbital energy is not same. There is a larger separation in between the orbital energy of one of the metal complex along with the another metal complex. But when the bond length difference is less, that means there will be a subtle energy difference in between these two metal complexes orbital. So when the energy separation is less, the bond length difference will be less. So in this condition, the electron can easily flow from one iron atomic complex to another iron atomic complex. But when the bond length difference is more, that means the energy difference in between the two metal complexes, it will be little bit larger. So when there will be little bit larger energy difference for electronic transfer, in this condition, we need to apply some activation energy. We need to apply some activation energy. So in first case, we don't need any types of activation energy for transfer of electron. But in second step, the bond length difference is more. That's why we need a activation energy for transfer of electron. So in both cases, electron transfer occurs. But in this second case, the electron transfer process is much more slow comparing to the previous one, because here the orbital energy of Fe3 plus and the orbital energy of Fe2 plus make a larger energy separation in between them. And here also the electron transfer occurs from T2G4 to T2G3. That means pi star transition to pi star transition occurs. Okay, you can make a readout here also. Fe3 plus OH2 bond length is 14 picometers smaller than the Fe2 plus OH2. This indicates that the energy level of the orbitals are not equal. And another example also you can consider. Here one is low spin, another is high spin. This is the first case we are discussing in between two inert complexes, and second step we are discussing about two labile complexes. And in third step we are discussing about inert and labile complex. The three different types of condition outer sphere electronic transfer can occur. And here also the electronic transfer process is very slow. So how can you predict the transfer process will be slow? From the bond length data. Here you can check definitely Fe3 plus and means its bond length will be smaller. Fe2 plus means its bond length will be more, but the difference is also more. Okay, that doesn't mean electron transfer occurs. Electron transfer occurs, but in a we need some activation energy. We need some activation energy or we need some input energy. Then electron transfer can occur. So here you can see T2G6, T2G5, T2G6. That means electron transfer will occur from this T2G to this T2G. So here definitely T2G5 electrons are present, but here the T2G orbital means three orbitals dxy, dyz, dx. These three orbitals called T2G orbital, and these three orbitals are occupied by six electrons. So when the incoming electron transfer occurs, it cannot occupy any pi symmetry. Okay, so that's why these two complexes are excited by some input energy, excited by some input energy, or we can apply some condition to make a electronic excitation inside the complex. So how can this electronic excitation occurs? You can check here. लिखा है देखो. It is mentioned that therefore it is necessary to excite the oxidation state before reaction can occur. So electronic excitation is necessary because the bond length is bigger. The bond length difference is bigger. We need more activation energy or we need more electronic excitation. So when electronic excitation occurs, you can see what will be the change in electronic configuration. Initially, the electronic configuration is T2G5, Eg2. When electronic excitation occurs, the electronic configuration will be changes to T2G6, Eg1. And here also you can see the electronic configuration changes to T2G5 and Eg1. Then now tell me, in this case, the electronic transfer can occur. Definitely, the electron transfer occur from this side to this side. Yes or no? Electron transfer कोठी हबो lower oxidation side to higher oxidation side. तो एठी मते को T2G6 ରୁ T2G5 के इलेक्ट्रॉन ट्रांसफर हे परिबो. एठी 5 अछि आउट इलेक्ट्रॉन से ऑक्युपाई कर परिबो. When the electron transfer occur from this side to this one, the electronic arrangement will be again T2G5, Eg1, and in lower side or in cobalt 3 plus side it will be T2G6, Eg1. So this is the electronic configuration of this one, cobalt 3 plus, and the upper one will be the electronic configuration of cobalt 2 plus. So you can see these types of electronic arrangement occurs. Further, they will rearrange to give different type of electronic configuration. T2G6 Eg0, T2G5 Eg2. It will become cobalt 3 plus, it will become cobalt 2 plus. So by initially by visualizing the electronic configuration of cobalt plus 2 and cobalt plus 3, you cannot say the electronic transfer occurs. But particularly here the electronic transfer occurs, and the electronic transfer is pi star electronic transfer. That means T2G6 to T2G5. Okay. So you can represent these types of electronic configuration by using the torque. In place of T2G, you can use pi star. In place of EG, you can use sigma star. So here pi star sigma star doesn't mean anti-bonding. It represents the pi orbital symmetry. That means T2G possesses pi orbital symmetry. That's why in place of T2G, you can write pi star. Okay. And the electronic transition occur from the pi star 6 to pi star 5. Then it will be pi star 5, it will be pi star 6. তার ক্ষেত্রে ইলেকট্রন আছে 7 6 plus 1 উপরে 5 plus 1 6. Then it rearranges to give pi star 6 sigma 0. It rearranges to give pi star 5 sigma star 2. Yes or no? And the cobalt 2 plus it will get converted to cobalt 3 plus, and this cobalt 3 plus it will get converted to cobalt 2 plus. Okay. So mostly the electronic transfer in outer sphere mechanism it will be faster when the two complexes are either high spin or either low spin. Okay, you can check another example also, cobalt similar example. Okay, so there is some short note type question he can ask. What is cross reaction? Details about the cross reactions. What is self exchange reaction? And there is a relationship in between cross reaction and self exchange reaction. This relationship is represented by Marcos-Horst theory. This one is most important. Okay. And this mathematical expression K12 it will be equal to K11 K22 K12 F the square root of this one. So by taking an example, you can easily understand what is K11, what is K22. Okay, suppose this iron water 6,3 plus complex, here we are taking ruthenium NH3 6,2 plus complex, it is forming this one and this one. Then this particular step, or I can say if you take only consider the iron complex Fe water 6,2 plus, and you are taking another Fe water 6,3 plus, then it will get converted to Fe water 6,3 plus, and it will get converted to Fe water 6,2 plus. Here the equilibrium constant I can represent it by K11. So when I am representing K11 and K22, that indicates a self exchange reaction. Here also, we are taking in the left hand side ruthenium complex, right hand side also ruthenium complex. And in this step, the equilibrium constant will be K22. And if you are taking two different types of complex, here the equilibrium constant will be K12. So to determining the K12, we need to know K11, we need to know K22, and there will be F. F is a statistical factor. So most of the time, X value will be provided, and F value will be provided. They will provide the value of F, and its amount will be mostly 0.85. So by putting this equilibrium constant value, we can calculate K12. Okay, so K12 is the equilibrium constant for the overall cross reaction. That means this reaction. And K11 is the rate constant for two self-exchange reactions. And this step and this step. So this mathematical expression is very, very important. They can ask theoretical question along with they can ask also numerical questions. Next we will discuss about the inner sphere mechanism. So in inner sphere mechanism, electron transfer occurs. One must be a oxidant, another must be a reductant. In both cases, in a outer sphere also, inner sphere also, then there will be electron transfer. One must be reductant, another must be oxidant. But here, the labile and inert nature is little bit restricted. That means here there will be strict condition that the complex or the reductant complex, it should be labile complex. It should be labile complex. Am I audible?
Yes, sir. Yes, sir.
Okay. So here The reductant complex will be a labile complex, and the oxidant complex must be a inert complex. And the inert complex possesses at least one ligand that can behave like a bridging ligand. That can behave like a bridging ligand, and this bridging ligand, it can make a coordination bond with the reductant's metal complex. With a reductant's metal complex, and it can form a precursor complex. It will form a precursor complex. That means in inert complex part, that means in which part? Oxidizing agent or oxidant or the inert complex part, there must be a bridging ligand. Bridging ligands means particularly we can say a ligand that can contain two lone pair of electrons, and the two lone pair electrons, it must have a good nucleophilicity that can it can donate to two metal atmosphere or two metallic complex. We cannot say water is a bridging ligand. Definitely water contains two lone pairs, but after donation of one lone pair electron, there will be a positive charge on water, so it cannot donate its next or another lone pair. But we can say OH minus, it can act as a good bridging ligand. We can say Cl minus, it can act as a good bridging ligand. Taurine minus, it can also act as a good bridging ligand. Okay, so if there is no bridging ligand, is mentioning that if there is no bridging ligand, then the electron transfer doesn't take place by inner sphere mechanism. So for inner sphere mechanism, there must be a bridging ligand in inert complex or in oxidant part. If bridging ligand is absent, we can say the electron transfer may take place by the outer sphere mechanism. Okay, and we can remember also inner sphere electron transfer reactions are faster than the similar reactions occurring through the outer sphere mechanism. Mostly the inner sphere electron transfers are faster compared to the outer sphere mechanism. Okay, so it's better to discuss about the examples. From examples, you can understood. So first we will discuss about the simple representation. This one will be the oxidant part OXX, and this one will be the reductant part ROH2. ROH2, that means it is containing a ligand, and this ligand is along reductant, and this will contain with a water molecule. Here there will be oxidant and there will be X ligand. So when the ligands, two metallic complexes of oxidant and reductant, it will react with each other. They will form a bond just like that. O means oxidant, X means bridging ligand, and R will be the reductant. And a water molecule from the reductant side, it will dissociate to outer side. That means in oxidant part, it contains the bridging ligand. Oxidant always contains the bridging ligand X, and this X will donate a pair of electrons towards the reductant, and it will form OXR. OXR, or this is called activation of precursor or formation of precursor complex. So from direct example also you can understood. You need to just wait. Here we can see direct example. We are taking chromium water 6, and we are taking cobalt plus 3, 5 ammonia and chlorine. And this chlorine will act as a bridging ligand. This chlorine will act as a bridging ligand. When it will form a precursor complex, you can see there will be removal of water molecule from this side. Here, how many water molecules will attach with chromium? 6. 1, 2, 3, 4, 5, 6. Here you can see 1, 2, 3, 4, 5. That means one water molecule it will be removed from this side. And this chlorine will donate a pair of another electron with this chromium, and this will form a precursor complex like that. That means the chlorine will be always available in the oxidant side. It will always available in the oxidant side. Just like here. Oxidant X are water molecule. This is the bridging ligand. They will form a complex like that. So after the formation of precursor complex, I can say the activation of precursor occurs or the electron transfer occurs from this side to this side. The electron transfer code is like chromium plus 2 to from cobalt plus 3. There it will be cobalt, then there will be a little bit error. It will be cobalt. Here you can see cobalt is present. This one will be also cobalt. So the electron transfer occurs from this chromium to cobalt. Chromium plus 2 to cobalt plus 3. So when electron transfer occurs, now the complex is similar, but now chromium is not chromium plus 2, it will be chromium plus 3. And this cobalt will be initially plus 3, but now this one will be cobalt plus 2 because chromium loses one electron and cobalt gains one electron. So cobalt in plus 2 case, it can behave like a labile bond with chlorine, but when cobalt will be cobalt plus 2 and it will be chromium plus 3, then this chromium atmosphere, it will be created just like a inert atmosphere. You can see here when this is chromium plus 2, that means it contains d electronic configuration. It contains d electronic d4 electronic configuration. When d4 electronic configuration is present, you can check the crystal field activation energy, it will be positive. But when it donates electron, now it will possesses D3 electronic configuration. Its oxidation state is plus 3. So D3 electronic configuration, the crystal field activation energy value is negative. That means chromium behaves like a inert complex. Now it will behave like a inert complex. When the complex will be inert, it cannot easily exchange its ligand, or I can say it cannot easily allow its ligands to participate with the another complex. Or in other sense, we can say the metal chlorine bond strength will increases comparing to cobalt Cl. So in further step, when dissociation occurs, this bond will not break. There will be breakage of cobalt Cl bond because here chromium Cl bond, it will just behave like a inert intact, or chromium form a inert configuration or inert atmosphere around itself. So when the further decomposition occurs, there will be a breaking of cobalt Cl bond, and we will get chromium water 6 2 plus complex. Okay, so this picture is from your e-ganacos material, or we can represent it by this particular representation: cobalt NH35Cl2 plus, chromium water 6 2 plus. This is a low spin complex, this is a high spin complex. Okay, and this final product will be this one: chromium water 5Cl2, and it will be cobalt water 6 2 plus. So how cobalt water 6 2 plus is forming? In the final step, in the final step here, we are adding 5 water further. We are adding 5 water molecules. This 5 water molecules, it will replace this 5 ammonia, and it will form this complex cobalt water 6 2 plus. That's why there is addition of 5 water 3 plus. Okay, you can check also. Initially, chromium 2 plus, it will form a labile complex. But when it will be chromium plus 3, you can check here. This is a inert complex. It is forming a inert complex. Or in theoretical language, if you want to predict or describe, you can represent also like that. Cobalt NH35Cl2 plus, chromium water 6 2 plus. You know water will dissociate from this one, and it will form a structure just like that: NH35, cobalt, Cl, chromium, water 5 4 plus. And in the second step, here from this complex, within this complex, electronic transfer occurs. So the first step is called the formation of a precursor complex, this one. And in the second step, the electronic transfer occurs from chromium 2 plus side to chromium cobalt plus 3. And this electronic transfer occurs through the bridging ligand. So the bridging ligand is necessary. For inner sphere mechanism, bridging ligand is necessary. That means electronic transfer occurs within a complex. A complex ভিতরে ହିଁ হচ্ছে. But outer sphere mechanism କ'ଣ ହଉଥିଲା? Will make intact two coordination sphere to each other. Through tunneling, one electron will transfer from this coordination sphere side to another coordination sphere side. And after transfer of electron, in the last step, there will be dissociation occurs in between the complex. So after the transfer of electron, now here the atmosphere will be little bit different. That means chromium electronic configuration will be chromium 3 plus. So chromium 3 plus means chromium possesses D3 electronic configuration. So when there will be D3 electronic configuration, you can check in your previous node, first class node, the crystal field activation energy will be negative. That means it will behave like a inert complex. Initially, you can see this chromium, this part will behave like a labile complex, but after electron transfer, this will behave like a inert complex. Because in the initial one, here D4 electrons are present, but here the electronic configuration will be D3. So D3 behaves like a inert complex. So when there will be inert complex, we cannot make breaking up chromium Cl bond. Rather, we choose to make dissociation of a bond from the labile side. That means in between cobalt and Cl. ओके तो नेक्स्ट टाइप जो बॉन्ड ब्रेकिंग होची से कोबाल्ट सीएल भीतर हबो कोन पे लाबाइल साइड रो हबो इनर साइड टा बॉन्ड स्ट्रेंथ कोन हबो स्ट्रॉन्गर फिर देखियो जो ब्रेकिंग हबो इट विल फॉर्म chromium Cl water pipe and cobalt NH3 52 plus. And in the next step we are adding one water molecule, further we are adding one water. Similarly we can add 5 water molecules that can replace all the ammonia and it will form cobalt water 6 2 plus complex like that. So this is called inner sphere electronic transfer mechanism. Okay, we can see this is a structure of your bridging ligand X, and one is from chromium 3 side, another is cobalt plus 3. The electron transfer will occur from chromium side to cobalt side through the bridging ligand. So you need to remember the electron rate of electron transfer process will be follow the order chlorine minus, bromine minus, iodine minus. So in case of iodine minus, the electronic transfer will be more or larger the size of halide, faster will be the rate of electron transfer. Or in other sense, we can say if the polarizability of the bridging ligand will be more, then the electronic transfer process it will be faster. Okay, you can read out here also. In bridging complex, the halide is more polarized by the higher charged cation. If the halide is polarized, the induced dipole moment is developed. Induced dipole moment developed means in the bridging ligand, one side will be occupied by the positive charge and another side will be occupied by the negative charge. Then it will behave just like a dipole. Dipole means one side will be positive containing positive charge, another side will contain the negative charge. So out of these four halides it can behave like a bridging ligand. Out of these four bridging ligands, in case of iodine, the electron transfer process it will be faster. Okay, so this is all about by outer sphere mechanism and inner sphere mechanism. Next, we will discuss about the energy processes, or particularly the about the pigments, hemoglobin, myoglobin, how it is helpful in the human or animal blood cell. Okay, so the red pigment of the human or any animal blood are intensely colored. It may be sometimes it may be red colored. Okay, so particularly this red color is due to presence of some pigments. So in human body, particularly the red blood color, is due to the presence of hemoglobin, particularly the hemoglobin. That's why this is called the pigments of life. That means it's offering the color to a blood. Without hemoglobin, or I can say the color will be different. It cannot be red color. So the hemoglobin, it will act just like a oxygen carrier, and it is helpful for the energy processes. That means it carries the oxygen from one part of body to the cell of the body. Okay, so its role is just like hemoglobin. I can say this will be oxygen carrier. When I'm saying myoglobin, it is the oxygen storage. It can storage lot of oxygen, and during the energy requirement process, it is offering the oxygen to the carbohydrate, and they can make dissociation within them, and they can form some energy, and this energy will utilized in the biological processes. So it may be hemoglobin or it may be myoglobin in any pigment. Iron is present in its formula, chemical formula. Iron is present. That means iron is essential in the coloring of coloring of blood. Along with it is also essential in other activities also. So the different types of pigments in which iron is present, we can discuss one by one. So today we will discuss about hemoglobin and myoglobin. The role of hemoglobin is oxygen carrier, and the role of myoglobin is oxygen storage. And in other proteins, iron sulfur protein also, iron can be a part of its chemical formula, part of its chemical constituents. You can see in cytochrome also iron is present, in reduction also iron is present. Okay, in protein codes ferritin transferrin ସେଠି iron present ରହୁଛି, in certain enzymes also that is required in nitrogen fixation. There also iron is essential. Okay, and some other enzymes, we can say aldehyde oxidase, peroxidase, catalase, succinic dehydrogenase. In there also iron is essential. So first we will discuss about the hemoglobin. So when I am saying hemoglobin, particularly 14 to 16 gram is necessary for males in this particular concentration range, and for female the range is 13 to 15. Okay, this is the general thing, but now we will discuss about its structures and its role. So this is the molecular weight of hemoglobin. You can see in the screen. This is the molecular weight of the hemoglobin, and its role just like an oxygen carrier in animals. Okay. So in most of the animals, it is the pigment to provide the color of blood. So the color of blood, it is due to the presence of hemoglobin. So the color of blood is red if hemoglobin is present with oxygen. In the absence of oxygen, hemoglobin can show the color of the blood is blue. That means for the color of blood, not only hemoglobin is present, along with oxygen also present. So if oxygen is present along with hemoglobin, the blood color will be red. So without oxygen, the blood color can be blue also. So, why these types of red color appears in the blood? This is particularly due to the pi-star transition inside the hemoglobin structure. When you will see the hemoglobin structure, there are so many pi bonds are present, and these pi bonds will transfer from one part of the complex to another part, and there is electronic transition occurs from one pi orbital to another pi star orbital. And this electronic transition, due to this electronic transition, the red color appears. Okay, so hemoglobin, first thing, it contains a quaternary structure, or we can say it contains four subunits within itself. Hemoglobin ভিতরে কোন আছে চারটা subunit আছে. That means we can divide hemoglobin into four parts. Or I can say hemoglobin means, we can say two terms, hemi and globin. Hemi means we can in next after 5 minutes I'll show you the hemi structure, or I can say one hemi unit, and globin means there will be protein chain. There will be protein chain. Okay, and this is the structure of hemi group. Hemi group means this will be a ring ligand. This will be a cyclic ring ligand, and cyclic ring ligand is called porphyrin ring. This one, when metal will present with this porphyrin ring or metal will inserted within the porphyrin ring, we can say this is a metalloporphyrin. And this particularly this metalloporphyrin is called one hemi unit. This is called one hemi unit. Okay, and this right hand side part is this is the hemoglobin structure. This is the hemoglobin structure. So this porphyrin part or this hemi group here we can see the yellow circles, and the iron 2 plus part will be the middle one. Yes or no? And these circles it is connected by each other through these chains. These types of chains. These are called the protein chains. These are called the protein chains, or I can say this globin part. Globin means these are the proteins. So these are the globins or protein chains, and this part will be the yellow circle along with this ball, little bit ball, small ball. This complete part is called hemi, and this part is called globin. That's why this is called hemoglobin. And this hemoglobin it is a quaternary structure. That means in one hemoglobin there will be four subunits are present like that, and these four subunits, or I can say the four hemigroups, it is connected with each other by the globin chain. By the globin chain. That means how many globin protein chains are necessary to form a one hemoglobin. We can say four globin chains and four hemisubunits. And this is the structure of one hemisubunit. And this one hemisubunit it is present here. It is representing this yellow circle part. And this small ball part will be the iron 2 plus structure. And these are the protein chains, just like one amino acid connected with another amino acid. Okay. So this protein chain, I can say this will be alpha chain 1, this will be alpha chain 2, and this is beta chain 1, this is called beta chain 2. So what is the difference between alpha chain and beta chain? In alpha chain sequence, 141 amino acids are present. In beta chain sequence, 146 amino acids are present. So according to different types of sequence, we can name one will be alpha, another will be beta. You can see another structure also. This is hemistructure, and this hemistructure representing the circle, and the middle red ball will be the Fe2 plus structure. And this thicker lines, and these types of thicker lines, blue color means it is representing the beta chain, and this types of violet color type thick lines it will represent the alpha chain. Okay, so you can see carefully one hemi-unit structure. You can see carefully this one hemi-unit structure. In one hemi-unit, we can see here four pyral rings are present. This is one pyral ring. This is another pyral ring. This is another pyral ring, and this is another pyral ring. And these pyral rings are connected by a CH group, ethylenic group. Here one ethylenic group is present. Here one ethylenic group is present. Here one ethylenic group is present, and here one ethylenic. So here, for conjugation, you can calculate how many pi bonds are present. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11. Okay. So here the extensive conjugation occurs just like that. And this pi bond, this cannot participate in extensive conjugation. It is present outside. Okay. Or in some side group or substitute group. So these only these 11 pi bonds, these are participating in extensive conjugation throughout the ring. So 11 means how many electrons? 22. Can it satisfy the Huckel rule? 22. 22 means 4n plus 2. It's satisfying the Huckel rule. That means it will behave like a aromatic ring. This one. And also remember that this aromatic ring or this porphyrin ring, it is just like a rigid ring. That means inside this cyclic ligand, there will be some specific size of cavity. So within this specific size of cavity, some few metallic atoms it can fit inside them. Not all the metallic atoms it can fit inside the cyclic ligands. Or I can say this porphyrin ring forming a rigid structure. Yes or no? This is forming a rigid structure. If we discuss about the crown ether complex, you may have heard about the crown ether, cyclodextrin. These are the flexible ligands. That means that can contain small metals also, and sometimes that can contain larger metal also. But here the cavity size is specific. That means some few metals, iron, nickel, or one to two other metal, it can fit inside it. Not all the metals. Okay. So before going to more discussion on hemoglobin, we know a little bit about its structure. What is the role of hemoglobin in body or in our human body biological processes? We can read out this one, this circle. Suppose in starting, we can say the oxygen enters into our body through the lungs or gills. When you're taking oxygen through the nose, it enters to our lungs or gills. Gills means in cases of fishes. Okay, so particularly I can say first if you take oxygen, it enters into the lungs. So in lungs, hemoglobin pigments are present. So first hemoglobin pick up the oxygen in the exterior blood. The exterior blood. Definitely in lungs also the hemoglobin initially catches the oxygen. That means the blood cells present inside the lung, it catches the oxygen through its hemoglobin pigment. Then the hemoglobin carries this oxygen through the blood towards our muscle, towards our muscle. And when it enters or reaches at the muscle tissue, it delivers the oxygen. First, it will bind with oxygen inside the blood when it passes through the lungs. Then again, it carries this oxygen from lungs to muscle tissue, and at muscle tissue, it delivers the oxygen. It delivers the oxygen. So, oxygen binding capacity and oxygen releasing capacity that will depends upon a particular factor, and this factor is called partial pressure. So, in lungs, the condition is like that. There, the partial pressure of oxygen is very high. So in high partial pressure, hemoglobin binds with oxygen. But in muscle tissue, the condition is like that. There, the partial pressure of oxygen is very low. That means there, in muscle tissue, the availability of oxygen is very low. That's why it delivers the oxygen. So it depends upon particularly the partial pressure condition. So the oxygen that will deliver at muscle tissue, it will picks up by myoglobin and it stores the oxygen. Secondly, when it will release the oxygen, when there will be a reaction with carbohydrate. When your body needs energy, it releases the oxygen, and this oxygen this will react with carbohydrate or we can see glucose. Okay, and there will be decomposition of this compound occurs. Glucose it will form carbon dioxide and water along with energy. Yes, sir. Then in next step, this CO2 it will react with water in presence of an enzyme. Next class we will discuss about this particular enzyme, carbonic anhydrase. This is a zinc enzyme. So in presence of this zinc enzyme, it will get converted to H plus HCO3 minus. First it will get converted to acid, carbonic acid, and again I can say this carbonic acid it is a composition of two ions, one H plus ion and this another anion. So what is the need of carbonic anhydrase? So the carbon dioxide will be highly toxic. If it is directly bind with the blood, then it will be harmful, very, very harmful for the oxygen. Yes or no? Or another thing is that the carbon dioxide binding tendency with our blood is very high comparative to the oxygen with our blood, because carbon dioxide is a strong ligand. It will behave just like a strong ligand, and oxygen is a moderate ligand. So oxygen it can bind in a little bit lower rate comparing to carbon dioxide with our body. That means hemoglobin it can easily pick oxygen, and comparing to carbon dioxide, if I say carbon dioxide in presence of carbon dioxide, its binding tendency with hemoglobin will be much more better, because carbon dioxide is stronger ligand. So our body is designed such that this carbon dioxide it cannot isolated here in this condition. It suddenly get converted to this H plus and HCO3 minus ion. And this HCO3 minus it will carried by the our blood serum, and again it is goes to the lungs. Okay. Again in lungs it will react with H plus, it will get converted to carbonic acid, and this carbonic acid further decomposed to give water and CO2. So again, through nose we are exhales the carbon dioxide in respiration process. So this is the complete biological cycle of the entry of oxygen in our body and how we are deliver the carbon dioxide. And through this cycle, inside this mechanism, the role of hemoglobin is much more important. Okay, so basically the role of hemoglobin is it is act as a oxygen carrier. It will act as a oxygen carrier. And we need to remember these points. That is the function of hemoglobin. Hemoglobin is a oxygen carrier. Also, it is a carbon dioxide carrier. But due to presence of carbonic anhydride zinc enzyme, this carbon dioxide cannot remain in an isolated condition. It is suddenly get converted to H2CO3 or H plus HCO3 minus ion. Hemoglobin maintains the shape of the red blood cells. Okay, so you need to just remember all these things. So hemoglobin is a composition of hemi and globin. One hemoglobin's molecular formula is this one C34 S32 N4O2 Fe. Okay, and one pyrrole ring it is get connected with another by a methane group, or this is called single bond CH double bond. That means this part is explaining about this part. This one C double bond C single bond. So the pyrrole rings are connected by this group, methane group, single bond CH, double bond. And hemoglobin consists of four subunits, each having one polypeptide chain or globin chain. Okay. So there are four globin chains are present. Out of these four, two are alpha chains and two are the beta chains. And the alpha chain contains 141 amino acids and beta chain contains 146 amino acids. And this is the complete structure of hemoglobin. You can see this is one hemi group. It is get connected with one chain, and this chain is connected to another hemoglobin, and from another hemoglobin another chain is connected to other hemoglobin, other hemi group, and from here one chain is connected to there. So there are four globin chains are required. Two are alpha, two are beta, and this single unit is called hemi group. This red part is called globin chain. That's why this is called combination of hemi with globin. That's why it is called hemoglobin. So when we are discussing about hemoglobin, there are two particular terms we need to remember: deoxyhemoglobin and oxyhemoglobin. So this is the condition of deoxyhemoglobin, when hemoglobin doesn't carry oxygen. যেତେବେଳେ hemoglobin oxygen carry করনি কি oxygen সাথে bind ହେଇକି ରହୁନି ତାକୁ କଣ କହିପାରିବା deoxyhemoglobin. And so another condition when hemoglobin binds with oxygen. So particularly hemoglobin binds with oxygen means particularly which metal will binds with oxygen, this Fe metal. So in this structure, you can see in the plane, iron is connected with 4 nitrogen atom. That means there will be no site of attack in the plane. So when a oxygen will make attachment with Fe2 plus, that attachment it can make from above the plane or below the plane. That means it can attack on above the plane or below the plane. The two sides are available, above the plane and below the plane, because the plane of this paper, or I can say the plane of this symmetry, it is coordinated or surrounded by other nitrogen atoms. So here we can say this is an amino acid of globin chain. It is connected with the iron atom, or making with the interaction with the iron atom, and the iron atom particularly doesn't present inside the on the particular plane. It is present little bit above the plane. It is present little bit above the plane. When its electronic configuration is Fe2 plus, Fe2 plus, and condition is high spin. So when I am saying Fe2 plus high spin, that means its electronic arrangement will be like that. T2G, this is the T2G orbital, and this is the EG orbital. Whose energy is more? EG orbital's energy is more. So when I am saying Fe2, that means it is a high spin condition. Fe2 high spin, D4, D5, and again D6. ଏଇଠି ଆଉ ଗୋଟେ ଇଲେକ୍ଟ୍ରନ୍ ପଳେଇ ଆସିବ। You can set it to G, EG. That means its electronic configuration will be like that. But in this condition, the Fe2 ionic size, metal ionic size is little bit more. When Fe2 plus in high spin condition or deoxyhemoglobin condition, it is available. It is form a dome types of structure. Why it will form a dome type structure? Because Fe2 to metallic size is much more bigger, little bit bigger. And you know previously the porphyrin ring and hemi ring, this cavity size it is fixed. When there will be some oversized metal, then it cannot injected inside the cyclic ligand. It will present little bit above the ligand. It will form just like this structure. Or I can say this will form a dome type structure. So this is the condition of deoxyhemoglobin. When oxygen, it will binds with the iron from above the plane. The A side oxygen will bind color. Then here the atmosphere of Fe2 will change. It will get converted from Fe2 high spin to Fe2 low spin. So when Fe2 low spin occurs, the electronic configuration will be like that. Eg and T2G. So these 6 electrons, it will occupy inside the T2G orbital. When the 6 electrons occupy the within the 3 orbital, T2G orbital, but here the 6 electron, out of the 6, 4 will occupy the T2G and 2 will occupy the EG. That's why the size is little bit bigger. But when the 6 orbitals will occupy within 3 orbital, its size will decreases. So suddenly, when Fe2 plus it will combine with oxygen, its atmosphere will change. It gets converted to Fe2 high spin to Fe2 low spin. So when Fe2 low spin is available, its size is little bit lesser or smaller comparing to Fe2 high spin. So in this condition, now Fe can get shifted towards the plane. Now it cannot form a dome type structure. In this condition, Fe will comes towards this plane or comes towards the cavity of the wing. When it can form a dome structure, when Fe2 is in a high spin condition or it is not coordinated with the oxygen molecule. But when it is coordinated with the oxygen molecule, its atmosphere will change. It will get converted to Fe2 low spin, and this Fe it will move towards or tilted towards the plane of the cyclic ring. Okay, so when oxygen will combine with the iron 2 metal, this condition is called oxyhemoglobin. So in oxyhemoglobin, iron atmosphere is Fe2 low spin. Fe2 low spin. When Fe2 low spin occurs, here you can see the iron will get shifted towards this plane. This is the structure one subunit of hemoglobin. This is one hemoglobin. Sorry, this is one hemigroup. It is connected with one oxygen molecule, and in the lower side it is connected with a histidine amino acid. And this oxygen molecule, it is also connected with a histidine amino acid. So this histidine that is present below the plane, this is called proximal histidine, and this histidine present on the upper side or through this side, the oxygen binding occurs. This is called distal histidine. Here the histidine is same, histidine is present. Here also the same histidine is present. But here oxygen also making a coordination with the iron. And along with the oxygen coordination, on another side of oxygen atom, it is also making a coordination with the this histidine. Okay, that's why this histidine is called distal histidine. This histidine is called proximal histidine. That means through proximal histidine side, oxygen binding doesn't occur. Through distal histidine side, oxygen binding occurs. And further, this histidine will be connected with another amino acid, it will connected with another amino acid, and it will form a protein chain, or I can say globin chain, like that. So always remember the oxygen, it binds with the metal complex Fe2 plus, along with on another side, it can also coordinate it with the another histidine. And this histidine is called distal histidine. ତାକୁ ଆମେ କ'ଣ କହିବା distal histidine କହିବା. So hemoglobin, myoglobin structure is very, very important along with its function. So what is the difference in between hemoglobin and myoglobin? There will be not much more difference in structure in between hemoglobin and myoglobin. That means hemoglobin contains 4 subunits. If I say myoglobin, this one subunit is called myoglobin. If I can say hemoglobin has quaternary structure like that, that means 4 hemi units are present. But in myoglobin, one hemi unit is present along with a single globin chain. Okay, that's why hemoglobin is called a quaternary structure. But you cannot say myoglobin possesses quaternary structure. Okay, so representation of myoglobin is MB, and representation of hemoglobin is HB. You can read out here also. Myoglobin is the monomer of hemoglobin that contains Fe2 ion plus ion in active site. So comparing to hemoglobin, myoglobin has a higher affinity for oxygen. Okay, but myoglobins are particularly present in the muscle tissue. It is not present in the lungs. So in muscle tissue, particularly the partial pressure is low. That's why hemoglobin in this condition, hemoglobin can release the oxygen, and myoglobin has a higher affinity to bind with the oxygen in this particular condition. So the role of myoglobin is to bind with the oxygen, and the role of hemoglobin is to carry the oxygen. You can see the structure of myoglobin. This is one hemistructure, and this is a single protein chain. And the red circle is the cyclic ring, and the yellow ball inside this is the Fe2 plus. And these little bit structures, these are the histidine. One is proximal histidine, another is distal histidine. So on the distal histidine side, oxygen will bind with the iron 2 plus complex along with this upper histidine. So one of the effect is called, most important effect is called cooperative effect. That means in a hemoglobin, four number of hemigroups are present. If one hemigroup binds with the oxygen, Fe2 plus ion, it will favor a condition to bind the oxygen with the second unit. This is called the cooperative effect. That means ଗୋଟେ ଆଇରନ୍ ଯଦି ଗୋଟେ ହିମୋଗ୍ଲୋବିନ୍ ରେ ଚାରିଟାଙ୍କ ସବ୍ ୟୁନିଟ୍ ଅଛି ସବୁଥିରେ ଚାରିଟାଙ୍କ ହେମି ଗ୍ରୁପ୍ ଅଛି ଗୋଟେ ହେମି ଗ୍ରୁପ୍ ସହିତ ଯଦି ଅକ୍ସିଜେନ୍ ବାଇଣ୍ଡିଂ ହେଲା this will helps to bind the next oxygen with the another iron atom ଗୋଟେ ଆଇରନ୍ ସହିତ ଅକ୍ସିଜେନ୍ ବାଇଣ୍ଡିଂ ହେଇଗଲେ ସେକେଣ୍ଡ ଟାଇମ୍ ଆଉ ଗୋଟେ ସବ୍ ୟୁନିଟ୍ ସହିତ ଯଦି ଅକ୍ସିଜେନ୍ ବାଇଣ୍ଡିଂ ହବ ତାର ଟେଣ୍ଡେନ୍ସିଟା କ'ଣ ହେଇଯିବ ବଢିଯିବ ବଢିଯିବା ଟେଣ୍ଡେନ୍ସି କ'ଣ କୁହାଯାଏ କୋଅପରେଟିଭ୍ ଇଫେକ୍ଟ ତମେ ଜଣଙ୍କ ସହିତ ଅକ୍ସିଜେନ୍ ବାଇଣ୍ଡିଂ ହେଲା. Suppose we can see the rate equation also. Simply I can say if the first hemoglobin is present, hemoglobin it contains four subunits. First one oxygen molecule will bind. Suppose if one oxygen molecule will bind, then definitely it will bind with out of these four subunits, it will bind with the any one subunit. And in this condition, suppose the rate of the reaction will be 10 to the power 2, and the product will be HbO2. And in second condition, that means hemoglobin O2 further reacts with another oxygen atom, and it is forming another further a molecule just like hemoglobin O2 2. Two oxygen molecules it will binds with the two subunits. ସେମିତି କେତେ ଯାଏଁ ହେଇପାରିବ hemoglobin O4 hemoglobin O2 4. That means in the first case, the rate constant or rate of reaction, you can say, if suppose it is 10 to the power 2, and in second case, the rate of reaction, it will be more. It can be 10 to the power 3 or 10 to the power 4. That means one binding of oxygen, it will make a favorable condition to bind with another oxygen atom. That means in second case, the rate of oxygen addition process is very, very fast. This is called cooperative effect. If one oxygen will bind with one subunit, it increases the ability of other subunit to make an approach with another oxygen molecule. This is called cooperative effect. And the cooperative effect structures may be present in your material. Okay. So whatever it may be, you can see structure. This one. This is proximal histidine. This is distal histidine. The oxygen molecule binds with above the plane with the iron atom. One side of oxygen atom it will bind with iron 3 plus, another oxygen atom it will bind with another histidine residue. Okay, so the oxygen binding with this histidine, that's why this histidine is named as distal histidine. This is called distal histidine. And below the plane there will be no oxygen binding, simply the nitrogen residue of histidine it will bind with the Fe3 plus. This is called proximal histidine. Okay, so cooperative effect is very very important. You can search in internet also. You will get its structures, these types of chemical reaction, first step, second step, third step. With the consecutive states, the rate of the reaction will increases gradually. Or I can set the addition of one oxygen molecule trigger for cooperative binding of another oxygen molecule with the another unit. Okay, so another effect is called Bohr effect. So Bohr's effect is called particularly the pH effect. So when the pH of the blood it will decreases or it goes to a acidic medium, then the binding tendency of oxygen with the hemoglobin decreases or affinity of hemoglobin along or myoglobin, you can say affinity of hemoglobin also or you can say affinity of myoglobin also, it decreases when the pH of the blood cells is low. At, in acidic medium, the binding tendency of hemoglobin and myoglobin with oxygen is very very low. This is called Bohr's effect. You can make a here there is a graph of Bohr's effect. Binding site, partial pressure of oxygen. If partial pressure of oxygen will increases, that means concentration of increases, then the binding tendency will also increases. Okay, and it is representing two curves. Particularly, we can see here this red curve. It is representing for the pH 7.6, and this black curve it is representing for the pH 7.4, and this blue curve it is representing the pH 7.2. In all cases, if you increase the partial pressure of oxygen, that means we can say simply with the oxygen concentration ବଢି ଯିବେ, then definitely hemoglobin ସହିତ oxygen concentration ର binding tendency କ'ଣ ହେବ ବଢି ଯିବ. But ମୁଁ କ'ଣ କରିବି ସେଭଳିଆ ତିନିଟା କଣ୍ଡିସନ ନେବି. ଗୋଟେ ରିଆକ୍ସନ ପରଫର୍ମ କରିବି in pH 7.6 ରେ, ଆଉ ଗୋଟେ pH 7.4 ରେ, ଆଉ ଗୋଟେ pH 7.2 ରେ. So you can check here when the pH is 7.2, that means little bit towards the acidic medium, the binding tendency is little bit lower. Yes or no? When pH is 7.4, the binding tendency is in between the middle. And when pH is more, the binding tendency is more. Yes or no? That means when pH is shifted towards the acidic medium, then the binding tendency of hemoglobin and myoglobin with the oxygen it will decreases. That is called Bohr's effect. That is called Bohr's effect. Okay. So particularly Bohr's effect works in this particular condition. In this particular the initial biological cycle. I already told you this one here. When hemoglobin comes towards the muscle tissue, in muscle tissue, particularly it will forming the carbonic acid. When the carbonic acid medium is forming, it forms a acidic medium. The hemoglobin binding tendency with oxygen it will releases. It will decreases. That's why hemoglobin releases the oxygen. It will catch up. It will be catched by myoglobin. Okay. So you need to just remember that when the pH will decreases, the binding tendency of hemoglobin with oxygen it will also decreases. That means the acidic medium is not good for binding tendency of hemoglobin. In acidic medium, or if the medium is little bit shifted towards the acidic part, then the binding tendency will decreases. Okay. So these two things they may ask questions, short notes also. Both effect and cooperative effect. These two things are very important. Okay. Thank you. If you have any doubt, you can ask.
Sir, how oxidation state is depends upon the bond length.
Oxidation state. Okay. Here also you can see. So ruthenium plus 2 plus NH3, ruthenium 3 plus NH3. Okay. So when the oxidation state of the metal will more, that indicates that here the positive charge density is little bit more. Positive charge density is little bit more. When the positive charge density will be little bit more, it can easily attract these ligands towards itself. So when the attraction will be more, definitely the distance in between the metal and ligand it will be very very less. That's why bond length is less. When the oxidation state is little bit less, that means positive charge density is less. When positive charge density is less, it cannot easily attract the ligands. Definitely it will attract, but comparing to this complex, here the attraction will be less because here the positive charge density is less. That's why the bond length will be more. Okay, or simply I can say ruthenium, a metal is also containing electron and a ligand is also containing electrons. But particularly in coordination chemistry, we can say the ligands containing the negative charge. That means out of metal and ligand, the electron density is more in ligand, and here the electron density is less because we are removing electrons, making it ruthenium 2 plus, making it ruthenium 3 plus. The electron amount is little bit less. তার মানে ruthenium রে proton comparatively proton और electron যদি ମୁଁ compare କରିବି ଏଠି କାହାର amount ଅଧିକା ଅଛି proton here mostly କଣ ଥିବ electron ଅଧିକା ଥିବ NH3 water that means it contains extra additional electron lone pair of electrons. That's why in crystal field postulate it is mentioned that metal will act as a point positive charge and the ligand will act as a point negative charge or point dipoles. Okay, just like electrostatic force of attraction, this will act like a negative charge and this will act like a point positive charge. If the
ସାର୍ ଏଇଟା
positive charge in
ଏଇଟା
okay, tell.
ହାଇ କମ୍ପ୍ଲେକ୍ସ ର ମଧ୍ୟ ସେମ୍ ରହିବ କି ସାର୍? ଗୋଟେ ଏକ୍ସାମ୍ପଲ
Hmm?
ଦେଇଥିଲେ ଯୋଉଟା କି ବଣ୍ଡ ଲେନ୍ଥ
Hmm.
ଡିଫରେନ୍ସ ଥିଲା, ମାନେ ହାଇ
Hmm.
ହାଇ ଅକ୍ସିଜେସନ ଷ୍ଟେଟ ରେ ହାଇ
Hmm.
ଥିଲା, ଲୋ ଅକ୍ସିଜେସନ ଷ୍ଟେଟ ରେ କମ ଥିଲା, ଗୋଟେ ଏକ୍ସାମ୍ପଲ
ନା ନା
ଦେଇଥିଲେ ହାଇ କମ୍ପ୍ଲେକ୍ସ।
ହାଇ ଅକ୍ସିଡେସନ ଷ୍ଟେଟ୍ ରେ ବଣ୍ଡ ଲେନ୍ଥ ଟା କଣ ରହିବ ଲେସ୍ ରହିବ ଦ୍ୟାଟ୍ ମିନ୍ସ ମେଟାଲ ଲିଗାଣ୍ଡ ବଣ୍ଡ ଟା ଷ୍ଟ୍ରଙ୍ଗ ହବ ବଣ୍ଡ ଲେନ୍ଥ ଟା କଣ ରହିବ ଲେସ୍ ରହିବ ସବୁବେଳେ ଆଉ ଲୋ ଅକ୍ସିଡେସନ ଷ୍ଟେଟ୍ ରେ କଣ ହବ ମେଟାଲ ଲିଗାଣ୍ଡ ବଣ୍ଡ ଟା କଣ ହବ ୱିକ୍ ହବ ବଣ୍ଡ ଲେନ୍ଥ ଟା କଣ ହବ ଅଧିକା ହବ ତ ବଣ୍ଡ ଷ୍ଟ୍ରେନ୍ଥ ଆଉ ବଣ୍ଡ ଲେନ୍ଥ ଇଟ୍ ଇଜ ଇନଭର୍ସଲି ପ୍ରୋପୋର୍ସନାଲ ଟୁ ଇଚ୍ ଅଦର
ନାଇଁ ସାର୍
ଏଠି
ଆଉ ଗୋଟେ
ଦେଖ
ଏକ୍ସାମ୍ପଲ ଦେଖୁଥିଲେ ଆପଣ
ହଁ ମୁଁ
ସାର୍,
ଦେଖୁଛି
ସେଠି ଡିଫରେନ୍ସ
ଏଠି
ଦେଇଥିଲା
ଦେଖୁଛି
ତ।
ଏଠି ଦେଖ 2 ପ୍ଲସ୍ ରେ କେତେ ଅଛି 214 214 3 ପ୍ଲସ୍ ରେ କେତେ ଅଛି 210 ଓକେ ଏଠି ବି ଦେଖିବ other any complex you can take any complex also. Here you see cobalt 2 plus କେତେ ଅଛି 211 ଏଠି କେତେ ଅଛି cobalt 3 plus କେତେ ଅଛି 193 ତା ମାନେ ଯୋଉଠି ହାୟର ଅକ୍ସିଡେସନ ଷ୍ଟେଟ୍ ଥିବ ସେଠି ବଣ୍ଡ ଲେନ୍ଥ ଟା କଣ ହବ ସବୁବେଳେ ଲେସ୍ ହବ ଏଠି ଦେଖ ହାୟର ଅକ୍ସିଡେସନ ଷ୍ଟେଟ୍ +3 ଏଠି କଣ ଆସିଛି 193 ଏଠି ଦେଖ ସେ ହାୟର ଅକ୍ସିଡେସନ ଷ୍ଟେଟ୍ କୋଉଟା ଏଠି ବଣ୍ଡ ଲେନ୍ଥ ଟା କଣ ଅଛି 210 ଏଠି କଣ ଅଛି 214
ହଁ
ମାନେ
ସାର୍
ଯୋଉଠି
ହେଲା,
ଅକ୍ସିଡେସନ
ନ୍ୟୁକ୍ଲିୟର ଚାର୍ଜ ଟା ବଢିଗଲା।
पॉजिटिव चार्ज डेंसिटी बढ़ीगला से इजिली लिगेंड को कला अट्रैक्ट करला प्लस 2 रे भी पढ़िथिबो प्लस 3 रे भी पढ़िथिबो देखथिबो पोस्टुलेट रे क्रिस्टल फील्ड स्टेबिलिटी क्रिस्टल फील्ड थ्योरी रे metal will act as a point positive charge and ligand will act as a point negative charge. Okay, just like electrostatic force of attraction metal ligand interaction in crystal field theory it is mentioned that it's just like a electrostatic force of attraction just or simply I can say coulombic force of interaction. A positive charge it will attract a negative charge. If positive charge density will it increases it can easily or strongly attract the ligand. So when a positive charge strongly attract the ligand then definitely the bond length will decreases. Okay, तो एठी हम जो इलेक्ट्रॉनिक ट्रांसफर रिएक्शन पढैछु एट सबुथरे देखिबो जोटे हायर ऑक्सीडेशन स्टेट थिबो तार बॉन्ड लेंथ ता कोन थिबो लेस थिबो जोटे लो ऑक्सीडेशन स्टेट थिबो तार बॉन्ड लेंथ ता कोन थिबो हाई थिबो बट इशू कोन तमे डिफरेंस देखियो पड़िबो एठी डिफरेंस केते आछि 4 गोटे जगह डिफरेंस कोन आसीबो अधिक आसीबो तो जोटे डिफरेंस अधिक आसुची ता माने এই কোবাল্ট 3 2 প্লস ର ଅର୍ବିଟାଲ ର ଏନର୍ଜି এই କୋବାଲ୍ଟ 3 ପ୍ଲସ୍ ର ଅର୍ବିଟାଲ ଏନର୍ଜି ଭିତରେ କଣ ହଉଛି ଟିକେ ଅଧିକା ଡିଫରେନ୍ସ ରହୁଛି ଟିକେ ଅଧିକା ଡିଫରେନ୍ସ ରହୁଛି ଯଦି ଦିଟାଙ୍କ ଅର୍ବିଟାଲ ଏନର୍ଜି ଭିତରେ ଅଧିକା ଡିଫରେନ୍ସ ରହିଲା ତାହେଲେ ଇଲେକ୍ଟ୍ରୋନିକ ଯୋଉ ଟ୍ରାନଜିସନ ହବ ଏଇ ମେଟାଲ କମ୍ପ୍ଲେକ୍ସ ରୁ ଏଇ ମେଟାଲ କମ୍ପ୍ଲେକ୍ସ କୁ ଯିବ ଇଟ ୱିଲ୍ ବି ଲିଟିଲ ବିଟ୍ ଡିଫିକଲ୍ଟ। ସୋ ଏଇ କେସରେ କ'ଣ ହୁଏ ନା ଆମକୁ କିଛି ଆକ୍ଟିଭେସନ ଏନର୍ଜି ସପୋର୍ଟ ପ୍ରୋଭାଇଡ କରିବାକୁ ପଡେ। ଦ୍ୟାଟ୍ସ ୱାଇ ଏଇ ରିଆକ୍ସନ ଗୁରା କ'ଣ ଥାଏ ସ୍ଲୋ ଥାଏ। ଯେତେବେଳେ ବଣ୍ଡ ଲେନ୍ଥ ଡିଫରେନ୍ସଟା ଅଧିକା ହବ। ଯେତେବେଳେ ବଣ୍ଡ ଲେନ୍ଥ ଡିଫରେନ୍ସଟା କମ୍ ହବ ଜାଣିବ ଯେ କ'ଣ ହବ ତାଙ୍କ ଅର୍ବିଟାଲ ଏନର୍ଜି ଗୁରା କ'ଣ ହବ ପାଖାପାଖି କ୍ଲୋଜଲି ଅଛନ୍ତି। ସେତେବେଳେ ଇଲେକ୍ଟ୍ରୋନିକ ଟ୍ରାନ୍ସଫରଟା କ'ଣ ହବ ଫାଷ୍ଟର ହବ। ତ ଏଠି ଦେଖ ଏଇ ଯୋଉ କମ୍ପ୍ଲେକ୍ସରେ ଏଠି 211 193 ଡିଫରେନ୍ସ କେତେ ଆସିଯାଉଛି 18 ପିକୋମିଟର ଆରାଉଣ୍ଡ 18 ପିକୋମିଟର। ବଟ୍ ଏଇ କେସରେ ରୁଥେନିୟମ କେସରେ ଏଠି ଦେଖ ଆଉ ସେମିତି ଫାଷ୍ଟରେ ଗୋଟେ ଏକ୍ସାମ୍ପଲ ପଢେଇଥିଲି ଦେଖିବ ଏଟାରେ ଫାଷ୍ଟ କେସରେ ଦେଖ Fe6 4 ମାଇନସ୍ Fe6 3 ମାଇନସ୍
Hello? Hello, sir.